let's talk about chloroplasts briefly. Chloroplasts capture energy from sunlight. They're large green organelles that are found in the cells of plants and algae, but not in animals or fungi. These organelles have an even more complex structure than mitochondria. In addition to their two surrounding membranes, they possess internal stacks of membranes containing the green pigment chlorophyll. You've heard of that, I'm sure. This is a chloroplast. These are chloroplasts in plant cells that capture the energy of sunlight. A single cell isolated from a leaf or flowering plant, seen in the light microscope, showing many green chloroplasts. A drawing of one of these chloroplasts showing the inner and outer membranes, as well as highly folded system of internal membranes containing the green chlorophyll molecules that absorb light energy. Chloroplasts carry out photosynthesis, trapping the energy of the sunlight in their chlorophyll molecules and using this energy to drive the manufacture of energy-rich sugar molecules. Cool. In this process, they release oxygen as a molecular byproduct. They release oxygen. That's what a plant does. Plant cells can then extract this stored chemical energy when they need it, in the same way animals do, by oxidizing these sugars and their breakdown products mainly in their mitochondria. Chloroplasts thus enable plants to get their energy directly from sunlight. They also allow plants to produce the food molecules and the oxygen that mitochondria use to generate chemical energy in the form of ATP. We'll talk about how these organelles work together in chapter 14. Like mitochondria, chloroplasts contain their own DNA, reproduce by dividing into two, and are thought to have evolved from bacteria, in this case from photosynthetic bacteria that also were engulfed by an early aerobic eukaryotic cell. All right, enough about plants. Although we'll do one last thing. Chloroplasts almost certainly evolved from engulfed photosynthetic bacteria. These bacteria are thought to have been taken up by early eukaryotic cells that already contained mitochondria, so a very similar concept here. Internal membranes create intracellular compartments with different functions. I like this word intracellular. You should use it when we're talking about inside of the cell versus extracellular. This is very important. Nuclei, mitochondria, and chloroplasts are not the only membrane-enclosed organelles inside eukaryotic cells. Okay, what else is there then, Mr. Textbook? The cytoplasm contains a profusion of other organelles that are surrounded by single membranes. Most of these structures are involved with the cell's ability to import raw materials, sounds like a business, and to export both useful substances and waste products that are produced by the cell, which we'll discuss in chapter 12. Here's the first one that they mentioned, which I've called out as very important. The endoplasmic reticulum is an irregular maze of interconnected spaces enclosed by a membrane. It is the site where the most cell membrane components, as well as materials destined for export from the cell are made. So it's like a factory. This organelle is enormously enlarged in cells that are specialized for the secretion of proteins. Stacks of flattened membrane-enclosed sacs constitute the Golgi apparatus, which modifies and packages molecules made in the endoplasmic reticulum that are destined to be either secreted from the cell or transported to another cell component. Lysosomes are small, irregular-shaped organelles in which intracellular digestion occurs, releasing nutrients from ingested food particles into the cytosol and breaking down unwanted molecules for either recycling within the cell or excretion from the cell. What's kind of interesting here is you have like a fractal representation. You have a cell that seems to have an input of raw materials and an output of waste. And this is at the cell level, which as we said is 2 to 5 micrograms, or micrometers, excuse me. But then we also have different organisms. I'm drawing my cat, who doesn't have a name yet. And the cat also basically has the same concept, right? The cat takes in internal raw materials and puts out waste. So we have this kind of like fractal relationship where at every dimension, as you zoom in, you sort of have the same concept. You can even argue that if we zoom out, all animals, not just cats, we kind of have an ecosystem that does the same thing, that takes in lots of raw materials and outputs lots of waste, not just excretion waste, but also wastes like pollution, right? So it's very interesting that you have this fractal relationship. Fractal is something that has the same nature no matter how many times you zoom in. If you want to learn more about fractals and the person that sort of discovered or invented them, Mandelbrot, I would highly encourage you to do so. So we're talking about lysosomes and how they break down different molecules and digest them, basically. And those are then recycled within the cell or excreted. Many of the large and small molecules within the cell are constantly being broken down and remade. Peroxisomes are small membrane-enclosed vesicles that provide a sequestered environment for a variety of reactions in which hydrogen peroxide is used to inactivate toxic molecules. That's why they're peroxisomes. Membranes also form many types of small transport vesicles that ferry materials between one membrane-enclosed organelle and another. All these membrane-enclosed organelles are highlighted here. So we're going to go through this in a second. All right, the endoplasmic reticulum produces many of the components of the cell. This is a diagram of the animal cell that shows the ER in green. Okay, look at how important the ER is, how much volume it takes. This is an electron micrograph. This is a schematic. This is the electron micrograph of a thin section of a mammalian pancreatic cell, so from the pancreas, that shows a small part of the ER, of which there are vast amounts in this cell type, which is specialized for protein secretion. That's in fact what the pancreas does, right? It secretes hormones. Note that the endoplasmic reticulum is continuous, and the membranes, or it's continuous with the membranes of the nuclear envelope. So here's the nucleus, here's the nuclear envelope, and you can see that you have this sort of like highway, basically, between the two. The black particles studding the region of the ER, you see these little black particles, and the nuclear envelope here are shown, are actually ribosomes, which translate RNA into proteins. Because of its appearance, the ribosome-coated ER is often called the rough ER, and that's different from the smooth ER, which doesn't have ribosomes. You can see parts of it that are smooth. All right, let's look at the Golgi apparatus. This is a little bit different from the ER. The Golgi apparatus is composed of a stack of flattened membrane-enclosed discs. Take a look at this schematic A and B. This is a schematic diagram of an animal cell with Golgi in red. Here's a more realistic drawing. Flattened discs, stacked flattened discs. Some of the vesicles seen nearby have pinched off to form the Golgi stack. Others are destined to fuse with it. Only one stack is shown here, but several can be present in a cell. This is an electron micrograph that shows the Golgi apparatus from a typical animal cell. And you can see it's kind of connected to the endoplasmic reticulum. And there's the nuclear envelope. All right, membrane-enclosed organelles are distributed throughout the eukaryotic cell cytoplasm. The various types of membrane-enclosed organelles shown in different colors are each specialized to perform a different function. The cytoplasm that fills the space outside of these organelles is called the cytosol. This is the cytosol, which contains the cytoplasm. Nuclear envelope, nucleus, transport vesicles, ER, Golgi, peroxisomes, mitochondria, and lysosome. So now you can kind of point to a cell and see what everything does. Pretty great. All right, let's get to something really important. The continual exchange of materials takes place between the ER, the Golgi, and lysosomes, the plasma membrane, and the outside of the cell, the extracellular environment. The exchange is mediated by transport vesicles that pinch off from the membrane. I want you to think about this concept of one organelle and fuse with another, like tiny soap bubbles that bud from and combine with other bubbles. At the surface of the cell, for example, portions of the plasma membrane tuck inward, that's the pinching process here, and pinch off to form vesicles that carry material captured from the external medium of the cell, a process called endocytosis. Animal cells can engulf very large particles or even entire foreign cells by endocytosis. In the reverse process called exocytosis, vesicles from the inside of the cell fuse with the plasma membrane and release their contents into the external medium. Most of the hormones and signal molecules that allow cells to communicate with one another are secreted from cells by exocytosis. How membrane-enclosed organelles move proteins and other molecules from place to place inside the eukaryotic cell is discussed in chapter 15. It's like a huge factory that needs like very detailed kind of instructions on how to ship everything from one place to another. All right, the cytosol is a concentrated aqueous gel of large and small molecules. This is a really good concept. If we were to strip the plasma membrane from a eukaryotic cell and remove all of its membrane-enclosed organelles, including the nucleus, ER, Golgi, mitochondria, chloroplasts, and so on, we'd be left with a cytosol. In other words, the cytosol is the part of the cytoplasm that is not contained within the intracellular membranes. In most cells, the cytosol is the single largest cell compartment. It contains a host of large and small molecules crowded together so closely that it behaves more like a gel than a liquid solution. And there's actually a really cool software program I'll show you from Scripps. It's called MESOSCALE. And you can see here, it's sort of a 3D model of a cell. So there's never really been software like this. And you can actually use this on the web. Don't remember exactly where. Let's see. Maybe I'll go here. Yeah, here it is. And so if I run this, I can actually, let's look at COVID. This is going to render kind of like a cross-section of a COVID virion particle. And you can sort of see what it looks like. And so oftentimes when you do these simulations, you do them a single protein at a time, and there's not really a good tool for visualizing the entire cell. So this tool was made for that purpose. Let's take a look at this tool, which is a bit more complex. It's rendering and downloading. It's very complex cell. This is a bacterial cell, or really a fungi, mycoplasma. And you can kind of see what they're talking about, that the cytoplasm is really, well, this is a bit more hollow than I would have expected, but really cool stuff. So you can play around with this software if you'd like. It's called MESOSCALE or Mesoscope, and it's made by Scripps. So anyway, the cytosol is more like a water-based gel. So eukaryotic cells engage in continual endocytosis and exocytosis to traffic things across their cell membrane. They import extracellular materials by endocytosis and secrete intracellular materials by exocytosis. Endocytosed material is first delivered to membrane-enclosed organelles called endosomes, which we didn't talk about yet, but really important kind of concept there. The cytosol is extremely crowded. This is sort of what the software I was looking at will make clear. The atomically detailed model of cytosol of 𝗕𝗏𝗃𝗁 is based on size and concentrations of 50 of its most abundant large molecules present in the bacterium. There are RNAs, proteins, and ribosomes in different colors. This is 25 nanometers, so pretty zoomed in. All right, so it's much more of a gel than a liquid. The cytosol is the site of many chemical reactions that are fundamental to the cell's existence. The early steps in the breakdown of nutrient molecules take place in the cytosol, for example, and it is here that most proteins are made by ribosomes. Okay. Can you give me a reason why it would be advantageous for eukaryotic cells to evolve elaborate internal membrane systems that allow them to import substances from the outside? I think that's fairly obvious. Let's talk about the cytoskeleton, and then we'll wrap up this chapter. The cytoskeleton is responsible for directed cell movements, and then we'll go on to our second Harvard textbook. Maybe we'll do linear algebra next, or maybe computer science. I don't know, whatever you guys want. The cytoskeleton is responsible for directed cell movements. The cytosol is not just a structureless soup of chemicals and organelles. Using an EM, one can see that in eukaryotic cells, that the cytosol is crisscrossed by long, fine filaments. Frequently, the filaments are seen to be anchored at one end of the plasma membrane or to radiate out from the central site adjacent to the nucleus. This system of protein filaments is called the cytoskeleton, and it's composed of 3 major filament types. This is pretty important. The thinnest of these filament types are actin filaments. They're abundant in all eukaryotic cells, but occur in especially large numbers inside muscle cells, where they serve as a central part of the machinery responsible for contraction. The thickest filaments in the cytosol are called microtubules, really important, because they have the form of minute hollow tubes. In dividing cells, they become reorganized into a spectacular array that helps pull the duplicated chromosomes apart and distribute them equally into the two daughter cells. Intermediate in thickness between actin and microtubules are the intermediate filaments. And I want you to think about that word filament, because it will come up again and again, which serve to strengthen most animal cells. So without these proteins, we would have cells that are just too floppy to really do much. These three types of filaments, together with other proteins that attach to them, form a system of girders, ropes, and motors that give the cell its mechanical strength, control its shape, and drives and guides its movements. That's really, really important. Without these, the description of the cell is completely incomplete. All right, the cytoskeleton is a network of protein filaments that can be seen crisscrossing the cytoplasm of eukaryotic cells. Three major types of filaments can be detected through fluorescent stains. So here's the actin, here's the microtubules, and here are intermediate filaments. Filaments are not found in the cytoplasm of cells with cell walls, such as plant cells. They have their own structure. Let's look at microtubules, which help segregate the chromosomes in a dividing animal cell. This is a TEM and schematic drawing here and here that show duplicated chromosomes attached to the microtubules of a mitotic spindle, which we'll talk about later on. When a cell divides, its nuclear envelope breaks down, and its DNA condenses into visible chromosomes, each of which has duplicated to form a pair of conjoined chromosomes that will be pulled apart into separate daughter cells by the spindle microtubules. But microtubules go well beyond just cell division. Because the cytoskeleton governs the internal organization of the cell as well as its external features, it is necessary to a plant cell, boxed in by a tough cell wall, as it is to an animal cell that freely bends, stretches, swims, or crawls. In a plant cell, organelles such as mitochondria are driven by the constant stream around the cell interior along cytoskeletal tracts, and in animal and plant cells alike depend on the cytoskeleton to separate their internal components into two daughter cells during cell division. The cytoskeleton's role in cell division may be its most ancient function. Even bacteria contain proteins that are distantly related to those that form the cytoskeletal elements involved in our cell division. In bacteria, these proteins also form filaments that play a part in cell division. We examined cytoskeleton in Chapter 17, discuss its role in cell division in 18, and review how it responds to signals from the outside of the cell in 16. So anyway, the cytosol is far from static. The cell interior is in constant motion. The cytoskeleton is a dynamic jungle of protein ropes that are continually being strung together and taken apart. Its filaments can assemble and then disappear in a matter of minutes. Motor proteins use the energy stored in molecules of ATP to trundle along these tracks and cables, carrying organelles and proteins throughout the cytoplasm and racing across the width of the cell in seconds. In addition, large and small molecules that fill every free space of the cell are knocked to and fro by random thermal motion, constantly colliding with one another and with other structures in the cell's crowded cytosol. Of course, neither the bustling nature of the cell's interior nor the details of the cell structure were appreciated when scientists first peered at cells in a microscope. Our knowledge of cell structure accumulated slowly. Let's look at the historical landmarks. 1665, we know that Hooke used a primitive microscope to look at cork, and he named them cells. 1674, Leeuwenhoek reports his discovery of protozoa. He sees bacteria for the first time. Later, Brown publishes his microscopic observation of orchids, describing the cell nucleus. Schleiden and Schwann proposed cell theory in 1839. 1857, Kolliker describes mitochondria. 1879, Fleming describes chromosome behavior and mitosis. 1881, Kyle, who's extremely important name in biology, and other histologists developed staining methods. Golgi sees the first season describes the Golgi apparatus in 1898, really crucial. Boveri links chromosomes to heredity by observing chromosome behavior during sexual reproduction. Palade, Porter, and Sjöstrand developed methods of EM that enable many intracellular structures to be seen for the first time. In one of the applications, Huxley shows that muscle contains arrays of protein filaments. 1957, Robertson describes the bilayer structure of the cell membrane, really important, seen for the first time in an EM. So that was not that long ago, if you think about it, about 70 years ago. 1960, Ken describes the first detailed protein structure, sperm whale myoglobin, to a resolution of just 0.2 nanometers. Very impressive. using X-ray crystallography. So that was 1960. And then Perutz proposes an even lower resolution structure for hemoglobin. 1965, a cell fractionation technique separates different organelles. 1968, Petran makes the first confocal microscope. 1970, Fry and Eddy used fluorescence antibodies, and that shows that molecules can get across the cell membrane. 1974, fluorescent antibodies used to stain the cytoskeleton. 1994, fairly recent, after I was born at least, they introduced GFP. GFP is a very important, very, very important innovation. 1990s, these folks developed techniques for super resolution fluorescence microscopy. So this is a relatively new technique. Let's go on to eukaryotic cells. Many may have originated as predators. Eukaryotic cells are typically 10 times the length and 1,000 times the volume of prokaryotic cells, although there's a huge size variation. They also possess a huge collection of features, a whole collection of features: nucleus, cytoskeleton, mitochondria, organelles that set them apart from bacteria and archaea. When and how eukaryotes evolved these systems remains something of a mystery. Although the eukaryotes, bacteria, and archaea must have diverged from one another very early in the history of life, eukaryotes did not acquire all their distinctive features at the same time. That makes sense. According to one theory, the ancestral eukaryotic cell was a predator that fed by capturing other cells. Such a way of life requires a large size, flexible membrane, and a cytoskeleton to help the cell move and eat. The nuclear compartment may have evolved to keep the DNA segregated. We should discuss the relative advantages and disadvantages of light and electron microscopy. Obviously, the disadvantage of electron is the preparation. The disadvantage of light is the resolution size. How could you best visualize a living skin cell, yeast, mitochondria, bacterium, and microtubule? All right, so the nuclear compartment evolved to keep the DNA separate from the hurly-burly, so as to allow more delicate and complex control of the way the cell reads out its genetic information. Such a primitive eukaryotic cell with nucleus and cytoskeleton was most likely the sort of cell that engulfed the free-living, oxygen-consuming bacteria that were likely the ancestors of the mitochondria. This partnership is thought to have been established 1.5 billion years ago, when the Earth's atmosphere first became rich in oxygen. A subset of these cells later acquired chloroplasts for engulfing photosynthetic bacteria. The likely history of these endosymbiotic events is illustrated above. That single-celled eukaryotes can prey upon and swallow other cells is born out of the behavior of many present-day protozoans, a class of free-living modal unicellular organisms. Protozoans, very important. Didinium, for example, is a large carnivorous, I don't know if carnivorous is the right word, protozoan, with a diameter of about 150 micrometers. Fairly large, 10 times that of the average human cell. It has a globular body encircled by two fringes of cilia, and its front end is flattened except for a single protrusion rather like a snout. Didinium swims at high speed by means of its beating cilia. This thing ugly, and it's eating this thing. When it encounters a suitable prey, usually another type of protozoan, it releases numerous small paralyzing darts from its snout region. Didinium then attaches to and devours the other cell, inverting like a hollow ball to engulf its victim, which can be almost as large as itself. Amazing. See how much more we have to go in this chapter. This is longer than I expected. Do we want to try a different textbook? This is just chapter 1. We're almost done.